Hi, welcome back to Sounds of Science. I'm Amy Myers, and today we're going to be talking about macromolecules, specifically organic macromolecules, and more specifically, four types of macromolecules. It's important for you as you go through this video that you understand you need to know these four macromolecules. You need to know their structure, you need to know their function, and you need to know examples of them. And we are going to do quite a bit with these macromolecules as we go through this unit. So I want to make sure that you understand them really well. Okay. First thing you need to know, what we're talking about here are organic molecules. Organic molecules just mean they have carbon. If they have carbon in them, they're likely an organic molecule. Some of the other elements that we might find in organic molecule includes things like hydrogen, oxygen, nitrogen, phosphorus, and sulfur. I really encourage you to come up with a mnemonic device that will help you memorize these. You may at some point be asked to determine which of the following molecules might be found in organic molecules, or which of the following atoms might be found in an organic molecule. And so what I want you to be able to do is I want you to be able to say, oh yeah, it's these specific elements. So this is the mnemonic that I use. No one can help our pupils study. So no one stands for nitrogen, can carbon, help hydrogen, are oxygen, pupils phosphorus, study sulfur. No one can help our pupils study. So hopefully that'll help you out. Carbon is a really, really cool atom. It has those 4 electrons in the outer shell. And because of that, they're actually able to bond to each other. Carbon can bond together. It can bond to other molecules. It can bond to another carbon and another molecule. So carbon's really kind of cool. And because of that, it comes up with these really super complex structures, either rings, like we see in glucose, or in long chains, like we see in fatty acids. But regardless, carbon is, as an element, allows us to create these really complex structures. And it really helps us have life on this earth as we know it. Without that, we're not going to see that. All right, let's talk monomers and polymers. When we talk about macromolecules, what you need to understand is that each macromolecule is going to be made of smaller molecules that have been put together. Those smaller molecules, we refer to them as monomers. Monomers are just going to be that basic subunit of the polymer that we're talking about, or of the macromolecule. When you take these little subunits and you start sticking them together, in a lot of cases, think of them like stringing beads together to make a necklace. But when you start sticking them together, they start bonding to each other. And when they bond to each other, they actually go through a process that we call dehydration synthesis. Water is released, much like this graphic is showing. Water gets released because a hydrogen and then a hydroxide are released together and they're going to form water. So it's called dehydration synthesis, even though water is being produced, because the polymer, as it's made, the monomers are actually dehydrating. They're releasing their water. The reverse process, when the polymers snap apart, is called hydrolysis. And essentially, that only is going to happen in the presence of water. It actually allows for the water to break down and then they'll rebond, as you can see in the picture that's been provided. So take a minute, look at these two pictures, dehydration synthesis and hydrolysis, and hopefully that will make sense as we move forward into our very first macromolecule. Carbohydrates. Carbohydrates are usually referred to as sugars. It could be simple sugars, it could be complex carbohydrates, complex sugars, could be starches. But regardless of what carbohydrate we're talking about, we're always going to see carbons, hydrogens, and oxygens in a 1 to 2 to 1 ratio. So, for instance, glucose. Glucose is 6 carbons to 12 hydrogens to 6 oxygens, a 1 to 2 to 1 ratio. We're going to see that in every carbohydrate we talk about. Typically, the whole purpose of carbohydrates is to release energy. This is quick energy. We get these glucoses, we break them down, we create ATP from them. We're using them for energy. Sometimes they're also used for structural support. They can also be used for protection. Um, Ironically, one of the carbohydrates that we'll talk about is cellulose, which is found in wood and trees. So, simple sugars, monosaccharides. The monosaccharides are the monomers of carbohydrates. Glucose is a monomer or a monosaccharide. So is fructose, which is that fruit sugar, and so is galactose. Um, those monomers get stuck together, and when they get put together, they're going to form a polymer, in this case called a polysaccharide. Polysaccharides are going to be things that are much more complicated or much more complex, things like starch. So the picture that I'm showing you here is 5 glucoses that have been stuck together through dehydration synthesis, right? And the starch itself is called glycogen. Glycogen actually hangs out in our blood, and our body starts to break it down when our blood sugar starts to drop. Some examples of carbohydrates: sweets, breads, sugars, honey, all of those things are going to be carbohydrates. You guys have heard of carbs before. You know what we're talking about when we say carbs. So just something to keep in mind. Our next macromolecule that we're going to talk about are the lipids. Lipids are things like oils and fats. We find a lot of them in butter, a lot of lipids in olive oil, and that sort of thing. They're also the waxy coatings on leaves are also a type of lipid. These guys are big. These are big molecules, and they're not going to be water soluble. This is why oil and water don't mix. Some of these lipids are also going to be steroids and hormones. They can function as a chemical messenger. And their other purpose is going to be long-term energy storage. So this is why individuals that take in lots and lots and lots of carbohydrates ultimately end up with lots of lipids. Those lipids are going to store that energy for the long term. And then eventually it can be released in times of famine, and you can get energy from it. So again, they can be steroids and hormones. They're chemical messengers, or they can be used for long-term energy storage. A couple of different types of lipids. We have saturated and unsaturated. You may have heard of saturated fats versus unsaturated fats. Truthfully, the only difference between the two, saturated fats do not have any carbon to carbon double bonds. So if you look at the picture where it says fatty acid structure, the monomer is a fatty acid. All those carbons have single bonds between them, no double lines. That's a saturated fat. It is not going to have those double bonds. So you have as many hydrogens as you possibly can have. Unsaturated is going to have at least one carbon to carbon double bond. And that means that it doesn't have all of the hydrogens it could possibly have, because one of those bonds that it would use for a hydrogen is being taken up as a double bond for a carbon. You can also have some of these fats have lots of double bonds, and we refer to those as polyunsaturated. So the picture that I'm showing you, the monomer is going to be known as a fatty acid. And typically when we talk about this, when we talk about the polymer, what we're going to be talking about is a collection of those fatty acids together. So a monoglyceride has one fatty acid and a glycerol. Triglycerides, which you may have heard of if you've ever talked about cholesterol or anything, your triglycerides are going to have 3 fatty acids together attached with the glycerol. They kind of look like an E, sort of shaped like an E with the fatty acids making up the E shape. So the monomers are fatty acids. Your polymers are going to be things like triglycerides. You can look for things like that. But most of the time, we just talk about them as being fats, um, oils, waxes, that sort of thing. All right, our third macromolecule is nucleic acid. So, nucleic acid is going to be made up of nucleotides. Nucleotide is a phosphate group, a pentose sugar, and a nitrogenous base. You guys have probably talked about DNA before and RNA, perhaps. That's what we're talking about here. That's our nucleic acids, our DNA and RNA. So, like I said, these nucleotides are made up of these three things, and these individual units form together to make our polymer, which is the whole nucleic acid. These are the genetic instructions for living things. Two types: RNA, which has the sugar ribose, DNA has the sugar deoxyribose. So, hopefully, this is a good introduction to nucleic acid. We're actually going to cover this a lot more in depth when we talk about protein synthesis. But here is just sort of your introduction to nucleic acids. And here we are at the last macromolecule, our proteins. Now, remember the last macromolecule I just talked about, the nucleic acids, as DNA, their job is to provide instructions for creating proteins. So this is what our DNA is making. This is what our nucleic acid is going to create. Our proteins are made up of chains of amino acids. So your monomer is your amino acid. Your polymer is that chain. We're going to call it a polypeptide chain. Amino acids truly end up looking like beads on a string, and they end up all attached together and eventually get folded up into the shape of the protein. So, amino acids themselves. We have about 20 different types of amino acids. They're all going to contain carbon, hydrogen, and oxygen. They're going to have an amino group on one end, which is that NH2, and a carboxyl group on the other, that's COOH. Okay, so all amino acids look the same. The difference is in the side chain. The side chain is what gives the amino acid its qualities, its characteristics. So the DNA, the nucleic acid, another macromolecule, tells the ribosome in the cell what amino acids to put into what order. This is going to create the proteins. And like I said, we're going to talk about protein synthesis in depth. We're going to cover it a lot more detail later. But just so that you're kind of seeing the introduction to this, this is actually referred to as the central dogma of biology. Huge concept in biology, extremely important. So, when I told you that the amino acids end up looking like a string of beads, you can see that on the picture on the right. And that's the primary protein structure. It eventually is going to twist into an alpha helix, or it'll form a pleated sheet. And then it's going to twist up even further and fold on itself, and then it's going to join with other folded amino acid structures. This is what gives us the protein. Proteins are actually really, really complex. They are really, really specifically shaped, because without that specific shape, it's not going to do its job. Proteins have jobs to do, and those jobs are structural, or it could be as an enzyme, something that causes reactions to occur. All of this is really important when we talk about how our bodies function. This is what gives us our traits. And if the shape is incorrect, then it's not going to work. They control reaction rates, they regulate cell processes, they form structures. If there is a change in the shape of the protein, it's a faulty protein, and it can result in a disorder. We get this usually by having a problem in the DNA. So this is where mutations come in. This is when we talk about genetic disorders. Okay, just because I mentioned them, chemical reactions is when one set of compounds gets changed into a different set of compounds. The starting compounds, we refer to those as reactants because they're going to be reacting. The ending compounds are the products because that's what's being produced. Okay, the most important reaction I want to talk about are reactions that involve enzymes. This is kind of a big deal. Life works because we have enzymes, which are a type of protein. Um, some reactions can't happen without some type of catalyst. A catalyst, in this case an enzyme, is not consumed, it's not used up in the reaction, but it does speed it up by allowing the reactants to come together. Therefore, they don't need as much energy. So, what you're seeing in this graph is that if you take your reactants and you put them together, they will eventually react. The reaction will eventually happen. But as you see by the red line, without an enzyme, it requires a ton of activation energy to get them started. Think of activation energy like if you are sitting at the top of a hill on a skateboard, right? As long as nothing happens, you're not moving. But if you get a little push, now you're flying down the hill, right? So that catalyst, that enzyme, is that push. It helps the reaction occur with less energy. You don't need as much energy. So the reaction goes faster or is more complete. Essentially, though, you end up in the same place. When we talk about enzymes, if they're shaped incorrectly, they're not going to work. They have to be shaped in such a way so that the substrates or the reactants are able to bind to the enzyme, just like in that picture on the bottom right. You can see how the substrate that those reactants are able to bind to the enzyme actually exerts, in this case, physical pressure and causes them to pop apart. This is what we talk about when it comes to enzymes and how enzymes work. It can also work in reverse. You can have the products come together and attach to each other. They can actually bond together because they're brought together by the enzyme. We're going to talk a ton about enzymes, and I'm hoping that this has sort of made sense. Um, if you are confused about enzymes, I'm going to put some more resources available to you to learn a little bit more about enzymes. So just stick with me. It's going to make sense before the end of the unit. So thanks for joining me. I look forward to seeing you next time.